morning, dear students. Today we start our class with amitosis. Last class, what we have learned? We learned about cell cycle, what are the checkpoints, all those things we have learned in the last class. Then coming to amitosis, which all organisms we can see amitosis? We can see this process especially in prokaryotes as a means of asexual reproduction. Look this word amitosis. A means absent. What is absent? Mitosis is absent. So it is a means of asexual reproduction in the case of prokaryotes like bacteria and protozoans. What is the process involved in this one? First, the nucleus. Suppose this is the cell, this is the nucleus. First the nucleus elongates, that's the process, elongates. Then what happened? Then it furrowed and divide into two nucleus. Divide into two nucleus. I will show that. It become two nucleus. And followed by that, the cell undergoes division by a process called cytokinesis. Furrowed and divide and form two daughter cells. And here the problem is that as there is the formation of mitotic spindle is not taking place, the distribution of the chromosomes to the daughter cells are not equal, unequal distribution. That is the difference when compared with the mitosis. Okay, is it clear? And this type of process, process of cell division is in prokaryote like bacteria, protozoans, and as an example, we can write binary fission. Specifically, binary fission in amoeba is a typical example of amitosis. The nuclear division happens by karyokinesis. And the cell division is happening by the process called cytokinesis. Okay. And that process we can see in this slide. Show that. Here, nucleus is elongating, it's dividing, and later the cell undergoes cytokinesis to form two daughter cells. Next, we are going to learn about mitosis. This is a main process, main division process that every cell need to pass through to increase the number of cells. First, we need to understand the peculiarities of mitosis. It occurs during M phase of the cell cycle, and it involves so many stages that you will learn later. And during this mitosis, the contents of the parent cell is equally distributed among the newly formed daughter cells. And also during that cell division, during different rounds of cell division, that cell, the parent cell is maintaining a constant chromosome number. So, even if a cell undergoes several rounds of division, then also its chromosome number remains constant. And during the mitosis stage, there is no transcription or translation. Why? We know that during mitosis, the chromosome is in its higher level of condensation. Different levels of chromosome condensation we already have learned in the former chapter. So, I hope that you all know what are the levels of that condensation. So, because the chromosome is in the higher level of condensation, it can't promote any transcription or translation. So, there is no scope for that. Next, we are going to learn about different stages of the mitosis. Different stages are there. For memorizing all the stages easily, there is a code. It's AMAT. Prophase, Metaphase, Anaphase, Telophase. Very easy to memorize. And first Prophase, we are going to learn that. And here we can find one another phase, that is an intermediate stage. Prometaphase means a stage prior to the metaphase stage. Okay, we'll come to that later. First, prophase. What are the major changes happening during the prophase? Condensation of chromosome is one major change that will happen during prophase, and disappearance of nucleoli and nuclear envelope. Nucleoli and nuclear envelope both will disappear. Formation of microtubules of the spindle is another important change that happens during prophase. Then we are going to understand each type of change in more detail. First is chromosome condensation. In the last chapter itself, you understand clearly that in interphase, after the S phase, the chromatin start to condense, and by the onset of mitosis, it finished its condensation, and then it appears in the form of chromosome with the two sister chromatids. Okay, so that condensation is finished by the beginning of this prophase. And kinetochore formation. What is kinetochore? That we learned in the last chapter. Kinetochore is a proteinaceous plate-like structure forming above the centromere of the chromosome. That also finished by the beginning of prophase. And mitotic spindle formation also started by the beginning of prophase. What is mitotic spindle? It is a basket-like structure that helps in the pulling apart of chromosome during the later stages of mitosis. So its formation also need to begin by prophase. For understanding more about mitotic spindle, we need to understand some other points. Mitotic spindle is formed of microtubules, and that microtubules begin its formation. It begin to grow around the centrosome. Suppose this is the centrosome. Around the centrosome, microtubule begin to form, and in the beginning it appears like bristles, and in that stage that microtubules are called aster fibers. Microtubules that begins its growth around the centrosome in the form of small hair bristles. At that stage, microtubules are called aster fibers. Since microtubules origin or its assemblance happens or assemblance begins around the centrosome, that centrosome is called microtubule organizing center. Okay. Then we need to know more about the centrosome. The centrosome consists of two centrioles that is arranged right angles to each other, barrel shaped centriole. So in one centrosome, two centrioles are present, and that itself arranged right angles to each other. Each centrioles are barrel shaped, and also each one is composed of one centriole picture is shown here. Each centriole is composed of a circlet of 9 triplets. Circle of 9. 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 triplets. 3 units are there. So centriole, two centrioles are present in one centrosome, and one such centriole is composed of 9 triplets. That's about its organization of the centrosome, and that centrosome stands as a organizing center for microtubule that form the mitotic spindle, and that mitotic spindle helps in the movement of the chromosome during the later stages of mitosis. Okay, clear? That centrioles are very clearly shown here. Two centrioles arranged right angles to each other, and from this these two centrioles form one centrosome, and from the centrosome microtubules are emerging. In its initial stage it appears like bristles, and that stage it is called aster, and as it extends it forms a basket, attaches with the centromere of the chromosome, and helps in the pulling apart of chromosome in later prophase stages. Sorry, later mitosis stages. Okay, and for moving the chromosome to two poles, two centrosomes are required for each cell. And for that reason, during interphase, the centrosome undergoes duplication, and the duplicated centrosome start to migrate to two poles by the beginning of prophase, and by late prophase, it will reach two poles. And after that, from that centrosome, microtubules start to origin as acid fibers, and it then start to elongate to form the mitotic spindle. Next major change that happening during the prophase is that disassembly of the nuclear envelope. Nuclear envelope, we already have learned about the disassemblance of this nuclear envelope in the last chapter. So I think you all remembering about that. By the beginning of prophase, that nuclear envelope disassembled, and in what form it exists after that? In the form of small vesicles. And during that time, that endoplasmic reticulum, Golgi apparatus, all those things also got fragmented and exists in the form of vesicles. That these are the major changes that happens during prophase. This is the picture of prophase. Again, I wrote here the major changes happening during prophase. Nucleolus, nuclear envelope, both are disappearing. Then chromatin condensed. Then separation of the centrosome to two poles. Formation of mitotic spindle started. These are the major changes that happens during the prophase. Next stage is prometaphase. It is an intermediary stage just prior to the metaphase stage. Which are the major changes that is happening during the prometaphase? This stage begins immediately after when the nuclear envelope disassembly finished. And during this stage, the mitotic spindle's assemblance will complete. And also, chromosomes from the pole start to move towards the equator during this phase. Then we need to know some other points regarding the mitotic spindle. We already understand that mitotic spindle is composed of microtubules, and that microtubules are of different types, and we are going to understand now. First one is polar microtubules. Polar microtubules are those microtubules that emerge from one centrosome towards the equator, and it overlaps with its counterpart that emerged from the other pole. That is polar microtubule. What is its role? It exerts a pushing force on the two poles to keep the two poles in that position itself without to prevent coming closer. That is the role of the polar microtubule. It exerts a pushing force to keep the two poles stay apart itself. Then next one is kinetochore microtubules. Kinetochore microtubules, they are certain types of microtubules that emerge from centrosome, and it comes and bind with the kinetochore of the chromosome. 20 to 30 such microtubules come and bind with the kinetochore of one chromosome. What is its role? It exerts a pulling force on this chromosome towards the pole later in the mitotic stages, later mitotic stages. So that is kinetochore microtubules. Then last one is aster microtubules. Aster microtubules are shorter microtubules that radiate from the centrosome. What is its role? It helps to position the spindle apparatus inside the cell. Then all the spindle microtubules have their minus ends. Minus end and plus end are there for each microtubules. And it's all microtubules minus end faces or directed towards the centrosome and plus end directed towards the equator or towards the centrosome. Sorry, towards the chromosome. Okay. Next stage is metaphase. In this stage, all the chromosomes are correctly aligned to the equatorial plane with the help of this mitotic spindle. And in this stage, we need to understand about a chemical. Its name is colchicine. Sorry, colchicine. What is its role? What impact it is causing on cell? If the colchicine is applied to a cell, it leads to the disassemblance of the mitotic spindle. What will happen if the mitotic spindle disassembled? That we know that with the help of the mitotic spindle only the chromosomes are correctly moving to the poles, and as a result it is equally distributing among the daughter cells. So that will not happen. So that's the effect of colchicine. And there are two types of mitosis. We need to know. First one is astral mitosis and second one is anastral mitosis. In astral mitosis, mitosis occurs with the help of centrosomes and asters. But in the case of anastral, the mitosis will take place, but there is no centrosome or asters. This astral mitosis we can find in all animals, and the anastral mitosis is present in all higher plants. Next phase is anaphase. During this phase, the sister chromatids of the chromosome split apart and start to move towards the poles. How that movement occurs? Because of the shortening of the microtubule. Where that shortening starts, it starts in the plus end that attaches with the kinetochore of sister chromatids. How that shortening happen? Because of the dissociation of subunits of microtubule. Each microtubule is formed of tubulin subunit. As this tubulin subunit dissociate from the microtubule, its length gets shorter, and that shortening exerts a pulling force on this sister chromatids and help this that sister chromatids to move towards the pole. Okay, and that anaphase is classified to two, anaphase A and anaphase B, based on the type of the force that is helping the chromatids to move towards the poles. Anaphase A is telling that the movement of the sister chromatids to the poles occurs because of the shortening of the microtubules. Anaphase B explains that the poleward movement of the sister chromatids occurs because of the elongation of polar microtubule. How is elongation happens? Because of the addition of tubulin subunits to its plus end. I will show that. As its length increases, here also its length increases, they meet each other and exerts a pushing force each other. That pushing force exerts or helps the centrosome on each end to move further apart. And that time this aster microtubules touches the cell membrane. That these two things helps the centrosome to stay firm more apart to the two poles, and that results or that helps the chromosomes to move further towards the pole. So that is about the anaphase B. And last stage of mitosis, that is telophase. And the telophase begins immediately after the chromosomes reach at opposite poles. And after that, the cell begin to return to the interphase stage by achieving so many changes. Which are the changes? Nuclear envelope reassemble. The chromosome again start to dispersed and decondensed, and then two daughter nuclei are reconstituted. The endoplasmic reticulum and Golgi complex that are disintegrated and exist in the form of vesicles again reunite and reconstituted. These are the major changes that happen during the telophase. And next, which are the driving forces that helps in the mitotic movement of chromosome to the two poles? The main agents are two proteins, dynein and kinesin. Dynein and kinesin. I will write its name here. Dynein and kinesins. They are two motor proteins. They are helping the movement of sister chromatids to the pole by work in association with the microtubule. Next, the last phase in cell division, that is cytokinesis. By the end of telophase, we found that two daughter nuclei are formed in the parent cell, and for forming two daughter cell, the next thing is to divide the cell. That process is cytokinesis, and that cytokinesis is initiated by the inactivation of the factor MEF, that is metaphase promoting factor. We learned about this factor in the first class of cell cycle, so I am not going to explain about it further. And so that factor need to be inactivated, then only cytokinesis begin. Then I am going to explain the process of cytokinesis in animal cell and plant cells separately. First, I am explaining the cytokinesis in animal cell. Suppose this is an animal cell, and first a contractile ring is forming in what direction? Perpendicular to the mitotic spindle. Mitotic spindle is forming in this direction. Perpendicular to that contractile ring is forming. That contractile ring is formed of actin and myosin protein. And its contraction of this actin and myosin, it pulls the plasma membrane. Progressively towards inward, and because of that pulling force, the two cells pinches off to form two daughter cells. This is the process that happens in animal cell, the formation of that contractile ring. And in the case of plant cell, we know that the main difference is the presence of cell wall. Here a new cell wall begin to form between the two daughter cells. A new cell wall. Its formation begins in the form of a cell plate. From the center, and this cell plate gradually progresses towards the margin, towards the lateral wall, and to this cell plate, as it progresses, further other materials of cell wall like cellulose are added to this cell plate, and as it touches the lateral wall, it joins with that and form two cells. And from the Golgi that located nearby to this cell plate, it helps to form a new plasma membrane for these two cells. Here also some Golgi may present. It helps to form a new plasma membrane. Here in this way, two daughter cell can form separately. Two independent daughter cells can form in the case of plant cell. That is the process of cytokinesis. I think it's very clear to you. Next, we are going to see about the biological significance of mitosis. Mitosis helps in the distribution of genetic material equally among the daughter cells. And also it maintains the chromosome number through all cycles of cell division. And it helps in the rapid multiplication of the cell to facilitate growth. And through mitosis itself, the body is replacing the worn out cells. And it is the basis of asexual reproduction. Mitosis is the basis of asexual reproduction. Okay, with this we finish mitosis and today's section. Thank you.
I'm an icon, man. I'm a girl about a 10. I should fuck her in a Benz.